Good day everyone. Today's lecture is going to be about spectroscopic methods. And in my lecture I will focus on FTIR and Raman spectroscopy of microorganisms. The outline of the lecture is the following. First I will give some basic introduction to microbiology and cell chemistry. Then I will introduce to you Fourier transform infrared spectroscopy, then Raman spectroscopy, and at the end I will show you some examples of using FTIR and Raman in microbiology. So when giving introduction to microbiology, I want first to give a prescription for this term. So microbiology is a scientific area which is studying different type of microorganisms which have a cellular form, as for example viruses, which have unicellular or multicellular forms, as prokaryotes, that's example of bacteria, and unicellular and multicellular forms, that's yeast and filamentous fungi. So in the application, when describing application of spectroscopy in microbiology, I will focus on bacteria, yeast and filamentous fungi. When we study microorganisms, nearly in all cases we are interested in cell chemistry of these organisms. And when describing cell chemistry, we need to know what are the basic inorganic and organic building blocks of cells. So here on this slide you see the basic inorganic chemical blocks, that's hydrogen, carbon, oxygen, nitrogen, sulfur and phosphorus. So these inorganic blocks building several organic building blocks: hydrocarbons, polysaccharides and lipids, amino acids and proteins, and nucleic acid, RNA and DNA. So another important aspect we can study it's in cell chemistry, that's chemical bonds. So we know that all cells, all cell chemical components, they have two types of chemical bonds: covalent and non-covalent bonds. Covalent bonds, that could be single, double or triple bonds, they are formed by the sharing of a pair of electrons between adjacent atoms. Non-covalent bonds, as for example hydrogen bonds, van der Waals interactions, electrostatic interactions, that's so-called interaction time bonds. So now slowly I will move to the introduction of Fourier transform infrared spectroscopy. When we look at the electromagnetic spectrum, Fourier transform infrared spectroscopy using infrared region of electromagnetic spectrum, and to be more specific, it lies in the mid-infrared region. So when we are analyzing our sample in FTIR spectroscopy, we are probing molecular vibrations. That's actually, we are going back to the chemical bonds. So we know that all chemical bonds in molecules, they are vibrating at certain frequencies. And these chemical bonds are able to absorb light at characteristic frequencies. So when we are doing measurements in FTIR spectroscopy, we are probing absorption of infrared light at these characteristic frequencies, and at the end we get so-called chemical fingerprint of the sample. So what type of molecular vibrations could be probed by Fourier transform infrared spectroscopy? So here you see different type of molecular vibrations. So there are two big groups which we can two big groups of molecular vibrations we can probe, that's stretching and bending. So stretching, that's a type of the vibration along the line of the bond. And here we have symmetric and asymmetric stretching vibrations. Bending, that's the vibration perpendicular to the line of the bond, and here we have a bit more variation in the types of the vibrations. So we could have scissor type, rocks, twist or wave type vibrations. In addition, it has to be mentioned that in FTIR spectroscopy, we are probing single, double, and triple bonds. So the information about these bonds we can see on different spectral regions, as for example, single bonds we are detecting at the region between 800 and 1300 wave numbers. Double bond, it's between 1700 and 1900, and triple bonds between 2000 and 2300. So basic principle of the FTIR spectrometer, this is very basic and very schematic principle. So what is happening in FTIR spectrometer when we are measuring a sample? We have a light source, and the infrared light comes from the light source. It goes to the beam splitter. In the beam splitter, the beam is split to the two beams. One goes to fixed mirror and another one goes to movable mirror. And then the beam goes back to the beam splitter from both mirrors, and then it goes further to the sample chamber and then to the detector. There are two types of measurement modes in FTIR spectroscopy which we apply in microbiology. So it's transmission and attenuated total reflectance. So on the slide here you see a high throughput FTIR plate, silicon plate, which is filled in with samples. And in the transmission mode, the light is transmitted through the sample. In the attenuated total reflectance mode, we are using single reflection crystal, and we plate a sample on the crystal, and the light is reflected from the sample. The application of FTIR spectroscopy in microbiology usually very often is associated with so-called next-generation phenotyping. If we take a microbial cell, we know that all microbial cells have four main components: lipids, proteins, polysaccharides, and nucleic acids. And when measuring FTIR spectrum of the microorganism, we get all information about these components we get on the spectrum. So here you see example of one filamentous fungi measured at different cultivation time points, and then at different areas of the spectrum we see information about lipids, proteins, polysaccharides, and nucleic acids. So the infrared spectrum is called a fingerprint. So in microbiology we consider infrared spectrum as phenotypic biochemical fingerprint. If we look in detail at the spectrum, I would like to show you where you can see or where you can find all the information related to the four main biochemical components. So on different regions marked with orange color, as you can see on the slide, you see all peaks related to the lipids. So I would like to note a special attention for the areas between 3000 and 2800. That's the area related to the CH2 and CH3 stretching vibrations, which is indicating about the length of the lipid skeleton. The area, or basically it's a single peak area between 1800 and 1700. That's related to ester bond stretching. The next component is proteins. So the proteins we have basically in two areas of the spectrum, but the most often used area for the proteins, that's between 1700 and 1500. That's area giving us information about amide-1 and amide-2 bands. Then we also can obtain information about phosphate in the cells. Not all cells has a phosphate, but some cells, as for example cells of algae and filamentous fungi, there is quite high amount of the polyphosphate in the cells. And there is usually two areas responsible for these components. And polysaccharides. So as you can see, the infrared spectrum can be split it in many regions depending on which chemical component you're looking at. Another important aspect of FTIR spectroscopy, also one of the biggest advantages of this technology, is that this technology is measuring intact cells. So compared to all other methods used in analysis of different metabolites of cells, and where we need to apply routines and long-term consuming protocols, FTIR spectroscopy doesn't require really any special preparation of the sample. It doesn't require extraction of the components. So we are measuring cells in its intact form. So here on the slide you see a SEM image of one of the sample positions from the FTIR plate, and that's a film of the yeast cells. As you can see, they have a nice intact form, and the cells are not disintegrated. In addition to cells, FTIR spectroscopy in its both modes can be applied to measure substrates which are used for growing cells. And here we will be able to get information about the substrate consumption, but also about release of extracellular components. So on this slide I just want to show you the difference between two modes, attenuated total reflectance modes and high throughput mode when measuring substrate. So on A picture you see FTIR mode, and on B is HTS mode. And then, for example, in both of the modes we can measure glucose consumption or the concentration of the glucose in the media. But you can see that in FTIR mode we get higher signals we get from the water and very low signals from the glucose, while in the high throughput, in the transmission mode, we get very high signals from the glucose, still high signals from the water, but they are much less of them. So both modes can be used for measuring these components in the media. Another difference is also related to how we prepare a sample, and that's also contributes to what we see on the spectra. So for FTIR mode, we are measuring sample in the liquid form without drying, and that's actually why we see a lot of water and quite high signals from the water. But in HTS transmission mode, we are measuring samples in its dried form. So we are spotting first samples on the FTIR plate, and then after drying at room temperature, we are measuring. So that's why amount of the water is much less in the HTS spectrum. The next subtopic is related to Raman spectroscopy. So I will give short description to this technology as well. So the first, what someone should say about Raman spectroscopy, that this technology is complementary to IR spectroscopy. So it means that by using two techniques, we get complementary information. In Raman spectroscopy, radiation at certain frequencies is scattered by the molecule with shifts. So, and then we observe these frequency shifts, and these shifts are related to the vibrational changes in the molecule, which are associated with IR absorbance. So that's why we can say that in Raman spectroscopy we are measuring Raman scattering spectrum, which resembles IR absorbance spectrum. But if we compare IR and Raman spectroscopy, so then there are three main points what you should remember, that in IR spectroscopy we are measuring vibration modes, and the same we are measuring in Raman, but in IR spectroscopy we measuring change in dipole, but in Raman we are measuring change in polarizability. Also, you should remember that excitation of the molecule to the excited vibrational state, that's something we measure in IR, but in Raman we are measuring momentary distortions of the electrons distributed around the bond. So, how to measure microbial cells in FTIR and Raman spectroscopy? So, before doing the measurements, we need to grow or cultivate the cells. And here you can use different approaches. So, the most traditional one is the shake flasks. And currently, more and more in use is more high throughput approach by using microtiter plate systems. So I will like, so first we grow the plate, we grow microorganisms, and I would like to focus more on the micro cultivation systems, because that's something what is the most popular nowadays. Then we need to prepare samples for the FTIR spectroscopy. So basically we need to wash cells from the cultivation media. And here there is a possibility to use a robotic system for sample preparation. We have such a system in our lab. The system is very robust, so it's doing cell washing and also spotting samples on the FTIR plate. And the system is able to handle about 200 samples per hour. And after samples are dried on the FTIR plate, we measure in FTIR. So the throughput of the FTIR is about 60 samples per hour. Let's look a bit closer at the high throughput macrocultivation system, which can have a throughput up to 2,400 samples in the same cultivation run. The system consists of the following components. So the first components, that's the microtiter type plates, and here you see different type of the plates which can be used. It's 94, 24 and 6 well microtiter plates. They depend, the number of the wells depends on the volume you want to use. So the less number of wells, it will allow you to use higher volume per well. The second component is sandwich cover. So as you can see, every well in the microtiter plate is closed by the sandwich cover. And the sandwich cover consists of the several filters. So the filter on the bottom is a soft silicone, which is allowing to close every well separately, and by that eliminate any crosstalk between wells. Also, we have several microfiber filters, and then we have a stainless steel foil in between the filters, and we have a stainless steel lid on the top. It's important to say that for every well we have a hole in the filter. So basically, if we are using 24 well microtiter plates, we have 24 holes in the sandwich cover. So these holes, they are allowing to keep a stable oxygen and CO2 transfer rate when we are growing our cultures. And the microtiter plates can be mounted on the incubator shaking platform by using clamp system. So that's a specially developed system which allows to mount different type of the microtiter plates covered with the sandwich covers on the top of each other. So the system I described to you now was developed by Enzyme Screen company in Netherlands, and it has been used, and it's used now a lot for different type of screenings in microbiology and biotechnology. So now we are moving to look at examples of using FTIR and Raman in microbiology. And the first example is related to the screening of biotechnologically important microorganisms, and I will use example of oleaginous microorganisms for lipid production. Oleaginous microorganisms, that's the microorganisms which are able to accumulate high amount of lipids in their cells, and that's mainly yeast and filamentous fungi, and also algae. So here on the slide, you see two oleaginous filamentous fungi, Mucorsa sarciniloides and Mortarella isabellina. And we have taken spectra of these fungal cells after growing them 1 day and 12 days. So the blue spectrum is related for the 1 day and the red is related to the 12 days growth. And since we are looking at the lipids, so I would like that we focus on ester peak and CH2 and CH3 stretching peaks. So what we see after 1 day of growth, we see quite significant ester peak for Mucorsa sarciniloides, but still it's smaller than polysaccharide peaks and smaller than amide-1 and amide-2 peaks in proteins. Similar observation we get in Mortarella isabellina. But after 12 days of growth, we see significant changes. So we see that ester peak increases a lot and it's much higher than any of the protein peaks, and it's nearly high as them as peaks in polysaccharide region. We also observe significant increase in CH2 and CH3 stretches. The same, even more significant changes we get in this fungi, Mortarella isabellina, where the peak related to the ester bond is increased so much that it's even higher than polysaccharide related peaks. And we have a very significant increase in the CH2 and CH3 stretches. That's I have to notice that CO stretching peak, that's related to the total content of lipids in the cell. So it means that after 12 days of growth in Mortarella isabellina, we have mainly lipids in the cell. And similar could be observed in Mucorsa sarciniloides. The next example is how to use spectroscopy for monitoring actually cultivations of oleaginous organisms. So here you see two examples of using FTIR for monitoring glucose and substrate consumption. Here we compared FTIR versus high-performance liquid chromatography, which is considered as a reference method for the measuring glucose in the substrate. And we also looked at comparing FTIR and gas chromatography, which is used for measuring lipid content in the cell. So, as you can see, the result observed by the FTIR and HPLC, they have a quite high correlation. So we get exactly the same glucose consumption from both techniques, and the same we observe for GC and FTIR comparison. We observe the same lipid accumulation curves obtained from the GC and from the FTIR. So given the fact that FTIR is correlating to both techniques, it allows us to use this technology as a standalone method without involvement of the reference techniques. Another example of using both FTIR and Raman in microbiology is related to the identification of microorganisms. So very often when we isolate microorganisms, we need to identify them and evaluate if they're spoilage or if they're pathogens. So and then we are taking a spectra of the isolates, and then we are trying to build a so-called phenotypic tree, hierarchical tree, which should resemble the phylogenetic tree and should resemble all the phylogenetic levels, genus, species, and strain levels. The next example of using FTIR and Raman in microbiology is related to monitoring analytical processes, and here I will talk about lipid extraction. So as I mentioned to you before, so the reference method for the lipid extraction is gas chromatography. But in order to analyze lipids by gas chromatography, we need to extract them. So basically we start with the microbial biomass, and we need to use a set of the solvents to get a phase separation and to get the lipid extract. And that's something what we measure in gas chromatography. And residual biomass, that's the biomass with extracted lipids. So since the protocol is quite long, we can get a lot of variations which will affect our results and the reproducibility of our results. So we introduced a method how to apply spectroscopy for controlling your extraction. Because when you are getting a lipid extract, you are not sure if you got extracted all the lipids from the initial microbial biomass and that in the residual biomass has no lipids. But if we take a spectra of the microbial biomass, we take a spectra of the phase separation of different phases we get after applying solvents, and we get a spectra of the lipid extract and of the residual biomass. We and compare the spectra with each other by looking at the lipid areas of the spectra. We observe, or we can evaluate, if there is a leftover of lipids in the residual biomass or not. We also can observe the quality of our lipid extract and the presence of any impurities, for example, protein impurities, from the biomass during the extraction. So the next example is related to the using these technologies for characterizing or for studying some pathogenic bacteria. And here we have, I will talk about Listeria monocytogenes, which is a pathogenic bacteria, and in order to treat infections of the Listeria monocytogenes, we need to understand the antibiotic susceptibility of this bacteria. That's also due to the rising issue of their antibiotic resistance. So here we have been using antibiotic bacteriocin, and by doing both reference susceptibility analysis and spectroscopy, we were screening 88 different strains of Listeria monocytogenes, which were exposed to the bacteriocin SKSNP, and we were looking for finding a susceptibility gap towards this antibiotic, which varied actually between two groups of strains. Yeah, and the susceptibility gap was obtained both from the reference method and from the FTIR data. So, in conclusion, I want to mention that FTIR and Raman spectroscopy, both techniques allow biochemical phenotypic fingerprinting of cells. That's actually not only cells, but in general, any material we analyze. So also it's important to know that FTIR spectroscopy in a transmission mode could provide high throughput analysis of cells, and that's it could be used in screening studies and also identification studies. Both techniques are highly reproducible, and because they are measuring intact cells, so they have very simple preparation protocol. Then FTIR and Raman spectroscopy, they are complementary techniques, so it means that by using both techniques we are getting more complementary and more complete information about our sample. We need to perform a quite complex data analysis of the obtained spectra. Thank you for your attention.